Thank you, James, and thank you for the kind introduction. So, yes, as James was saying, my name is Sergio Latorre. I'm based in UCL, and I'm really excited to be here with you with this Get Genome seminar. Today, we are going to talk about the journey that is from Contigs to Clades. And this is particularly challenging for me because, as you can imagine, just from the title, we are going to touch about fungal population genomics. So we are going to go from all the journey from sequences, from raw sequences or assemblies, all the way through some meaningful insights into evolutionary biology. So bear with me if I start like jumping from methods to a little bit of evolutionary theory to some examples that I have been doing myself with the work and with my work on the rice blast fungals, which I'm gonna talk about today. Yeah. So let's start with this reality of modern genomics. And as you may know, sequencing costs have gone down really fast. In this chart from the NIH, they highlight the fact that sequence technology have gone faster than Moore's law, which measures the pace of computing technology. So if you notice this white line, this is the Moore's law, and that measure the pace where technology is keeping up with time. So if you notice the y-axis of this particular graph is log scale, and that means that every 2 years, if you see the ratio of this line, every 2 years, sequence and technology is doubling in pace. So what these authors want to reflect here is like sequencing technology is even going faster than the computing technology itself. And the reduction of the cost is halved every 2 years. If you take, for example, from the early 2000s till a couple of years ago. And of course, you can see this big drop when there was the transition from pure Sanger sequencing to the second wave of technology when we had sequencing, and further in the third wave of sequencing technology when we had Illumina sequencing. So all in all, this is pretty impressive, and costs have been massively dropped down, and it's really fast, the pace of sequencing. Another data that I want to bring on is this study, is the second plot. There is a meta study about the 1000 human repository, and they want to show that there is some bias in the researchers when they use legacy sequencing data. What I want to show instead is, of course, you should avoid very old sequencing data because it's full of noise and a little bit of bad quality, as we know nowadays. But I want to highlight is the fact that sequencing is not getting just cheaper, but it's getting better and better with the time. So what you can see is like this trend in improving the sequencing quality, regardless of the sequencing technology. So this is a little bit outdated, this study, but the message is clear. Sequencing is getting cheaper and is getting better by the time. And this is not just some exciting statistics, it makes real-time genomic monitoring like very feasible. So it means that nowadays you can go to the field, you can take a sample, extract DNA, do a library preparation, sequence it, assemble it in a matter of days. We're not talking from the old times of the Human Genome Consortia that took like almost decades, but we are talking about days. And this is very exciting time, and that puts like another layer of complexity, but also about like the computational challenges that all these data put on us. And that, of course, is a little bit of a game changer if you think about the perspective in agriculture and in the perspective of public health. And now, why focus on fungi? Many of our most important pathogens, including the one that I am studying, which is Magna porthuroiza, are predominantly haploid. And that is important because it simplifies very much the assumptions, but also the analysis that we can do with these raw material that are the sequences. But most importantly, because these fungi and many of the pathogens, either if you are a researcher on the field in agriculture or the human health, many of these pathogens have very small genomes. So just to give you an overview, with the typical output of one of these mini ion flow cells has roughly 15 gigabases. And with one flow cell, you can either sequence a quarter of a human genome, but instead you can sequence at a 30x coverage 12 very decent fungal genomes. And I'm saying 30x, which is pretty decent given the fact that they are haploid genomes. So we're talking about like real powerhouse, like it can fit in your pocket. I don't want to give more advertisement to particular companies, but the take on here is that we can have a really good quality and a lot of power in this new technology and these new devices. The flip side of many pathogens being haploid is that, as I was saying, it simplifies, but also you have to be very careful about their biology and about their physiological characteristics. As many of you may know, fungi genomics can be very challenging because of they have parasexuality, they can have anastomosis, there are high presence of repetitive regions, there is presence of disposable chromosome units that just recently we start to be more aware of those. We have presence of huge transposable elements, nowadays known as starship elements, and all these characteristics, which are like very particular from fungi genomics, make them a little bit challenging and particular if you want to inquire some of these workflows that are used for other organisms like humans, like flies, etc., etc. So it's true that we can borrow some of those techniques, some of those models, but always with caution and always putting the focus on the particularities of our particular system. And as the title suggested, I want to put the highlight on the population structure. For me, population structure is the foundation of almost everything we do in evolutionary genomics or evolutionary genetics, because first, it allows us to reconstruct demographic histories. We can see how species or particular lineages of that particular species have changed through time in terms of population sizes, how they have experienced demographic events like bottlenecks, like expansions, which are particularly often in agricultural intensification. Population structure also let us resolve evolutionary history, species boundaries, hybridization events, that some and many times in these fungal pathogens is very important because many of them have cryptic species. There is exchange of genes, and there are like these hybridization zones where we are not sure whether they belong to one species or the other. So evolutionary histories really gave us many insights to try to disentangle this complex biological evolutionary history of these organisms. We can detect new genetic diversity. We can track novel haplotypes or introductions from unsampled geographic sources, but always if we have this blueprint, if we have this framework, which are the populations. The population structure also help us to identify variants of concern. By knowing how populations are structured, we can pinpoint mutations linked to, for example, fungicide, resistance, virulence, etc., etc. This is more important after what we have experienced as humanity after COVID. The variants of concern are actually on the highlight of this type of programs that are aimed to monitor real-time outbreaks. And of course, it allows us to monitor pathogenic lineages in space and time. And coupled with what I was suggesting in the previous slides, now with this technology, we can use the power of this technology in order to real-time monitor outbreaks and diseases across both agriculture and health important pathogens. I want you to appreciate that when we suggest population structure is not just one particular step in the pipeline of analysis that as a researcher you want to do, but is probably the foundation, one of the most important foundations of your whole research if you want to inquire about the evolutionary history of a fungal or any other particular organism. But also, before looking at the data, let's revisit some classic framework. And I want to bring like the classics of Wright's about the evolution of Mendelian populations, which was like almost 1 century ago, when Wright was like advising the populations, the creation, the splitting, and the merge of populations as like some forces that were pulling apart or merging or blurring some of the individuals back in populations. First of all, let's say that mutations are, as you may know, are the raw material. They create variation. Mutations are either maintained, fixed in a population, or removed. Depending on they are going to be fixed or removed depending on how detrimental or deleterious they are. First for the individuals themselves, but also in the context of populations. So again, mutations are going to be fixed or removed from populations depending on how detrimental or beneficial they are. So for example, if a pathogen acquires a mutation that confers resistance to a fungicide, this population will thrive in crops, intensive pesticide usage, and the mutation is likely to rise in frequency in that population because it's beneficial. However, if you think about the same mutation happen in an ecosystem where pesticides, where fungicides are not being used, probably that particular mutation and the consequences of that mutation are going to behave a burden for the pathogen and for the population itself. It requires more the energy to maintain that particular allele, and because it's not used, it's going to behave like as a burden. So the same mutation can have different consequences depending on the type of organism, depending on the time of the selective pressures, and depending on the ecosystem in which this mutation is maintained and acquired. So the take on here is like mutations are beneficial or detrimental depending on their phenotype, on their phenotypic role, but also in the specific environment. So knowing that the mutations are the source of the raw materials to create populations, what are the forces that are at play? So one force is drift, or it could be seen also as the founder effect, which is very characteristic in fungal or pathogenic populations in general. The isolation of individuals or sets of individuals into new geographies, into new environments and hosts will allow these particular mutations to, as the name says, to drift away and increase in frequency. So just give enough time for these mutations to fix in the populations, and the individuals that have been isolated will drift away and they will create new populations. So this is very intuitive to think in a temporal or in a temporal scale process. Drift, mutations plus drift create populations. And those effects are stronger in very small populations because drift is stronger in small populations. But then you can see the opposite type of forces. If you think, for example, that after populations have been settled, if migration occur from one population to the other, that's the opposite force. So for example, imports of plant material that carry pathogens will of course force pathogen populations into new ecosystems when they can interact. They can recombine, they can exchange genetic material, and they can eventually replace the former local population. Think about, for example, the use of similar agricultural practices, the use of similar fungicides in different latitudes, in different environments. There are similar selective forces that push the appearance of similar alleles here and there. So there are like forces that with the same raw material that are the mutations can create forces, put them apart, like the drift and the founder effect, or can blurry the borders of those particular populations. Yeah, but I want you to highlight one particular message, and is that population differentiation is a continuous process. It's not a qualitative process, but it's a continuous process. It's a quantitative process, if you like. The clusters that we draw in order to make sense of the data are always cutoffs, are always thresholds that are arbitrary, and as researchers, we need to do it to try to make sense out of the data. They are useful, but let's not forget that they are simplifications of a very more complex reality about these forces that are a continuous process happening time by time and minute by minute. Okay, with this little bit of theory, let's jump straight to our protagonist, which is Magnapore theoriza. This is the blast fungals. What I want to say first is this is a multi-host pathogen, as you can see in the different pictures. It can infect plants like wheat, like rice, sorghum, triticum, setaria, triticum. Most of the cereals can be infected by Magnapore theoriza. And actually is one of the most devastating and most important agricultural pathogens. So the consequences can be devastating. This organism also has a fascinating biology. It is mostly asexual in the field, meaning that it reproduces mostly clonal. And as I told you, it is a multi-host pathogen, but most importantly, there is a tight association between its general population structure and the host that it infects. And you can appreciate that in the figure that I brought here. This is a quick tree made with very low resolution, but the main message of this particular tree is that there is a tight association between the plants represented by these different pictures and colors and the population structure of the pathogen. So look, for example, all these green colors have this very, very small branch in this particular clade that has been expanded from this point onwards. There's similar things happen for the wheat, which are in blue. And I must want to say that both rice and wheat are the most oversampled organisms on the field. So most of the research have been doing both in rice and wheat. And because of that, I want to focus on the rice lineage, which is like this green color that I show in the bottom. Just give me 1 second. So yes, I'm gonna focus in the rice lineage. So that what did we go? We revisited the framework of a 2020 paper that we did with some colleagues from the Sainsbury lab. And then we revisited this in the light of new samples. So back in 2020, we did like a meta-analysis in that we gathered hundreds of short read sequences and we put them together in order to try to verify what was the worldwide population structure of this particular pathogen. What I want to do right now is getting new data, and this new data comes from assemblies, high quality assemblies that were not sampled back in 2020, and test whether our assumptions and our framework of 2020 holds with the new data that we can see. So this is going to be a little bit of a workshop. I just want to guide you through some really quick and insightful analysis that you can do also with this type of data. So as I was saying, we took 20 high quality long read assemblies, which are high quality sequenced and assembled, and asked for different questions. Is there evidence of genetic differentiation across these samples? What are the characteristics of the populations? Is there evidence of geographic structure? What is the relationship between populations, mating strategy, and hosts? So let's see if we can stretch the power of these 20 samples to try to give at least insights in many of these questions. I want to say that, of course, many of these analyses, you can spend more time refining the analysis. So in a way, they're like quick and dirty analysis, but it's just like to try to show you what is the power of these sequences that can be produced real quick and relatively low budget. So first things first, we have always to assess the assembly quality. So the starting point here are the assemblies. Then we did some of the basic statistics, as many of you may know, in order to try to assess the quality. But this is also true if you start with raw sequences, regardless if they are short or long. There are many metrics or many quality statistics in place that you must consider when you assess the quality of your starting data. So we look at standard statistics like the number of contigs, which in an ideal scenario should match the number of chromosomes of your organism. In this particular case, we are talking about a fungi, which has repetitive regions, which even though we can have like long read sequence technology, still you struggle to assemble some of them. We are talking about a fungi which has accessory chromosomes, which by definition are not core chromosomes. Therefore, you never know what is the real number of chromosomes that you have in your particular sample. But a good quality sample should be considered something that is relatively close to your core chromosomes. So we look at standard statistics. We also looked at the total size of the assemblies. And again, you can see that there is some variation in the number of bases. It ranges from 4.3 megabases all the way to almost 5 megabases, which is the range that many of these ASCO can range. And on a side note, this is fantastic, the level of flexibility of these genomes. And you can imagine that many of this variation is attributed to these non-core chromosomes, which are the accessory chromosomal elements, or these huge transposition events that represent starship elements. So this is something incredible as a side note. But also you have some statistics like N50, which put the weight on the half of the mass of the distribution. So what you want to see here is relatively small numbers that you can, and small and compact numbers in your data set. So those numbers and those statistics looks reasonably well for starting assemblies. Of course, you can imagine that you can start also by end-to-end, telomere to telomere type of high quality assemblies. Most of the time we don't have that. So I want you to put like something very realistic that we can have. We have high quality, we have long read based assemblies, but it's not the perfect and ideal scenario. Something very important that you want to start with is, after looking at more technical statistics, looking at something more biological meaningful, which is the BUSCO, or the benchmarking sets of universal single copy ortholog. So this group of people have done something incredibly valuable, and is putting together a huge database on many organisms across the tree of life. And for the particular case of the type of fungi we are studying, we have a tailored database, which is the Sordiaromycetes, which represent, among others, this particular class of fungi that we are studying. So the idea here is that you are going to look for specific genes that must or should be present in any single fungal of this particular class. So an ideal scenario is that you have 100% of these particular genes, but of course, this is not an ideal world. So you want to be as close as possible to 100% of BUSCO scores. The results, as you can see, reasonably good. I would say quite good, because you can see that the complete BUSCO scores are above 98%, which is, we are probably missing, in the worst of the scenarios, 2% of these genes. And you can also spot real quick that one particular sample can have a little bit of a problem of duplication, because this is the raw statistic, but then you can see more ingrained type of metrics. For example, the complete single copy BUSCO or the complete duplicated BUSCO, and this is just a mirror image of one or the other, and that there is one particular sample with a little bit of duplication. And these things happening in reality, I would say you have to make a judgment call what is acceptable or unacceptable in any stage of your process, depending on the type of insights that you want to, and the type of analysis that you want to do. So in this particular case, I'm gonna do population structure, which are like genomic wide signals. So my judgment call is I will still include this particular sample, but if I want to pursue more downstream analysis, like pinpointing specific genes, looking for specific haplotypic diversity in some specific alleles, then I will be flagging this particular isolate and investigate further what is going on. It can be an artifact, most of the cases. It can be something interesting as well. Okay, so let's crack on population structure. And I want to present 4 different approaches or flavors of analysis, and can give really powerful insights. One is distance-based methods, and I'm going to be talking about quick methods that can work as preliminary analysis. I'm going to talk about dimension reduction analysis, specifically about PCA. I'm going to talk about ancestry-based analysis, specifically about what we know as admixture or structure type of analysis, and talking about phylogenies. So again, the starting point is those assemblies that we already made the quality control check. And then I'm gonna do a K-mer-based method. A K-mer-based method is gonna take advantage of the full representation of the dataset. One of the advantages in this type of methods is that they do not require reference. For those of you that have been dealing with mapping, with aligning to a particular reference, this is not a trivial task to do, because you have to be very thoughtful about to choose the reference genome. If there is no prior information or prior knowledge about your type of model organism or a model organism, you have to spend some time trying to choose the right one. You have to choose one that has gold standard characteristics. But on the other hand, you have this type of methods that are K-mer-based, which are really helpful in these situations. So what is this about? You take the whole assemblies and you dissect them, and you chop them in very small words, or namely K-mers. You put a hash of them, and this is information theory. So this is just like an abstract representation of every single K-mer, and then you have sets of representations of K-mers. What you can do is, if you have a set of K-mers represented with some hashes for sample A and the same for sample B, you can do very, very simple arithmetic operations. For example, finding what is the union and what is the intersection of those sets. And for those of you who remember a little bit about intersection and union, you can imagine that you can create distances out of those simple operations, better known as Jacquard distances. So this is a very computational, simple way to try to have a sense about genetic distances between two particular sets of genomes. One of the most used tools or bioinformatic tools for that is MASH, but there are many others out the market. And MASH it sacrifices a little bit of precision because it's just an estimation or a representation of the k-mers. So that means that it's not exhaustive. But there are like some other tools that are exhaustive and can have better results. Nevertheless, because I told you that this was a very preliminary analysis, so we used MASH. Then what we aim for is that creating this 20 by 20 symmetric matrix, 20 columns representing the 20 individuals that we have, plus against each other. As you can appreciate immediately, is this zero diagonal because it's comparing each individual with itself, so the distance is 0. But if you compare each organism versus the other, then you have a matrix of distances. And if you have a matrix of distances, you can put on top of that any clustering algorithm. Like in my case, I use neighbor joining. So I want to highlight a distinction here when I draw. Neighbor joining is a clustering algorithm that you can make on any meaningful distance matrix. In this case, is Jacquard distances. So this is only clustering algorithm. Please notice that we haven't put any biological or any evolutionary knowledge or theory in building this particular result, but it's just like pure information theory. This is just like pure distance methods, but we can have a visual representation of the clustering. And we can start drawing some conclusions about our dataset. For example, that we have 3 particular nodes in which some of the samples are clustering together, plus we have like some other individuals, which seems a little bit more disconnected from the rest of the dataset. So we can start like drafting some preliminary conclusions by this very quick type of analysis. But as I was telling you, the real deal is to call genotypes on your dataset. At the end of the days, you want to have confidence of the genotypes. And in order to call genotypes, you can imagine that because each assembly is unique by itself, you have a repertoire of many chromosomes. There are regions in one assembly that are not in the other. So you have to put some level of order among these assemblies. And this is done mostly by choosing one of your best, if you want, your best assembly, and try to align every other one to that particular. So there are like 2 main criteria that I would say you should pursue in order to select what is your reference genome or what is your reference assembly. One of them is like the statistics tell you what is the best assembly with the best numbers in terms of number of contigs, in terms of N50, in terms of the Busco completeness, as we saw. And this criteria is very important, but the other criteria is like what has been used in the field as a standard reference genome, because that might be stronger and that put a little bit more weight in what do you choose. Sometimes there is a compromise, but always be mindful about these pros and cons while choosing your reference genome. Once you have chosen, you can have many tools, like for example, Minimap, which is extremely fast and quick, and this is the one that I use for this particular seminar, but then you can see many others in the market as well. You must apply a very strict mapping quality filter because your starting point are namely high quality assemblies. So don't worry about strict mapping quality filters. And then after you have aligned each of the assemblies to the reference genome, then you can call genotypes. And at the end of the day, what you want to end up is with a SNP matrix that can be represented in many different formats. Some people are more familiar with VCF variant call format. There are some binary representations of this matrix, but at the end of the day, what you want to end up is the same coordinate system that identifies specific genotypes. One of the huge downsides of this type of approach is that you're using a single reference. And as I was saying before, in this particular fungi, those genomes are so flexible that you ended up by having a reference bias. This can be circumvented by a new pangenomic type of analysis, but for this particular talk, I'm going to choose one particular reference and I'm going to map to that, knowing in advance that I can suffer from some reference bias. But again, as I was saying before, I'm just like circumscribing this analysis to population structure, so the effect might be negligible in this type of analysis. Then we're going to go to dimension reduction type of analysis, or PCA analysis. So you have your SNP matrix and you have your genotypes. So you can imagine that each of the genotypes or each of the SNPs represents a feature. So you have a very complex matrix, and what PCA is all about is like reducing the complexity of thousands or even sometimes millions of SNPs into just 2 or 3 meaningful dimensions. And this is most of the time what in the PCAs. What you want to see is some level of clustering or not of your population. And what I can see immediately is that now it's revealing some level of clustering in my samples. So I have here 1, 2, 3, maybe 4 type of clusters. If I look at the two main principal components, if I look at the second or third component, I still see the second one cluster, another cluster here, another cluster there, and maybe some dispersion in something that in these first two components looked apparently as a cluster, but now it feels that it is spread out in the third component. So this type of analysis help you to draw some new conclusions about it. You can see a little bit more of a structure in this type of data. And because I was telling you that I know more or less the framework of this particular pathogen, I can assign specific labels to these particular clusters. Nevertheless, let's keep this in mind that if you take just the PCA as your to-go analysis, you ended up here with a clustering of 3 populations or maybe 4. There are some post-analysis that you can do in order to evaluate the robustness of these clusterings, for example, silhouette scores, k-means algorithms, but all of them are just like on top of the principal components. Now I want to present an orthogonal method which has a little bit more a model rationale behind it. So again, the starting point is the SNP data, the genotypes. What these particular algorithms try to do is to optimize Hardy-Weinberg equilibrium. So the way that they are going to do is you can imagine that you are going to assign theoretical ancestral populations to describe how your data clusters with those given ancestral populations. So for example, I can tell the program to imagine that there are like 2 ancestral populations, and therefore the program is going to split all the samples of my dataset into 2 theoretical populations, and then to 3, 4, 5, and so on and so forth. So the common practice is to iterate through a k equal to 2 to k's that goes 6, 7, depending on your dataset. So for example, this is what happened when I do the modeling with 2 ancestral populations. So I want to put in parallel the PCA because it's very meaningful to observe what is the relation between the one and the other. And what this mixture model is telling me is 5 of my samples are distinguished as one population, which represent this one that I color here in green, and the rest of them are mostly blue, which represent the one that were clustered in the left-hand side of the PC number 1. That makes sense because the PC number 1 should be the most powerful to tease apart the samples, right? And there are like some others here that even though they have mostly a blue component, they have a little bit of mixture with this green ancestry component, which I highlighted here where my laser point is showing. So this is the result with k equal 2, but as I was telling you, the idea is that you go iteratively k3, k4, and so on and so forth. So for example, when k equal 3, I have 3 ancestral populations, and the way that the program is telling me is my populations are splitted here, these 5 individuals, which are consistent with the same individuals that were classified before with k equal 2, are these individuals that I'm showing here. There are like 5 individuals in blue colors, which are these ones, and now there are like 5 individuals in red color, which are these individuals here. And the remaining 5 individuals are a mixture of the 3 ancestry components, which represents the individuals that were here. And then you can see the rationale and the logic of this type of modeling. If I equal k to 4, the picture is relatively similar. there is maintenance of these 3 particular subpopulations, while these 5 individuals get separated with a different ancestral component, and a single one of them seems like a little bit of a mixture. And if I keep going and further, for these 3 particular populations, the picture is the same, and what changes is the level of granularity and separation of these individuals. Now, think about the following. If I start increasing K to match the same number of individuals that I have in my dataset, the higher the better. Because at the end of the day, if I have K equal to 20, every single individual is going to classify apart from the other individual. So what's the point of doing this K? So there should be a manner to decide what is the optimal level of the ancestral components. And this is done by a cross-validation procedure, which is hiding part of your data and letting the model predict what is the hidden information and contrasting that predicted information with your real values. And this is what you know as the elbow shape type of plots, in which where you have the lowest point, it represents the lower type of error of your predictions or the cross-validation error. And this is the K that is mostly better explaining better your data set. So the results here is like choosing a K of 3, it represents better my populations. So at the end, I have a conclusion, and is that my data set is represented by 3 different populations plus a set of individuals that are mixing ancestries somehow. And this is where also your biological and physiological insight should come in place, because it could be very tempting to say or to suggest that there is some level of admixture happening in these individuals. Indeed, they seem like they are mixing from different sources to make up their genetic makeup. But what we know about this particular fungal is that they are mostly asexual, as I was telling you. So what I want to do now is creating a visual representation of what is the genetic relation among them, and on top of that, putting what I know about the mating type. So what I have used here is a network, which is a particular method that can handle mating, that can handle recombination, because I must say that when you do phylogenetic frameworks, you should be very cautious, because some people used to throw phylogenetic trees on anything. And of course, if you use a tool, if you use a program, the program is going to spit you out of a result. But one of the 1.1 main assumptions of phylogenetic reconstructions is that you don't have recombination. However, there are some other alternative methods or algorithms that can handle recombination, because recombination is nothing but just like the distortion of the topologies, because recombination breaks the shared ancestry and the shared evolutionary history between alleles, between SNPs. So what I'm representing here is a tree in which recombination can be described by the reticulations here. And when you have like more thin lines, the less recombination or less topological discordance you have. Again, I can see a representation of my main 3 different colors, green, blue, and red. But what I'm plotting here on top of this information is the mating type. And the mating type, I just did it with a series of blasts, looking and fishing for mating type 1 or mating type 2. And what I can see is like in these 3 particular lineages, there is one unique type of mating type. Here, mating type 1, here only mating type 2, and here only mating type 2, while in these 5 other individuals that seem a little bit far away from the rest of the data, there is a mixture of the 2 mating types. So what I can suggest, or what these results suggest, is like there are like 3 clonal lineages that have a unique mating type. So recombination is likely playing like a minor role, or is not playing a role at all in these particular lineages. So they are clonal. They don't have anyone to mate with. So if I know that they are clonal, then I can pursue a more phylogenetic approach. And I'm going to go a little bit faster because the time is of essence to let you make some questions. And what I did here is like, because I am not violating the assumption of clonality, I removed all these individuals that are not in clonal lineages, and I computed a pure phylogeny. What I see in this phylogeny is, again, the 3 clusters, green, blue, and red. But this is very important because that gives you certainty about the relationships between samples. and this is known as the bootstrap of the phylogeny. So most of the time you're going to have like very high bootstrap support values if you don't have if you have a clean data set. But in some cases you have different values. For example, this value here is extremely low, and this could be actually interesting because that can suggest that the topology in this particular phylogeny is discordant for many reasons. It can be allelic share, it can be genetic share, it can be introgression, it can be horizontal G transfer. It can open the possibilities to try to draft many hypotheses that are interested on this. Something else that I did here, as you can see, I put the labels of the countries of origin of these isolates, and you can start drawing some conclusions. For example, this particular green lineage is only found in samples coming from Italy and from Japan, while the blue and the red one are found mostly in Africa and some other latitudes like in the Americas, for example. I know that, for example, in Italy and in Japan, predominantly people grow Japonica type of rice, for those of you who know a little bit of crops of rice, while both in Africa and in America, there is like some predominance of the indica type of varieties. So again, I can start drawing some hypotheses that I can test further down in the road. So at the end of the day, I have 4 different orthogonal methods that in conjunction, they give me a pretty good understanding about what is the population structure, and they allow me to draft some hypotheses. A glimpse of the type of analysis that you can do afterwards is asking about the genetic diversity levels in these particular populations. Once I have drafted and I have a pretty good grasp of my populations, then I can ask, have the clonal lineages accumulated high levels of genetic diversity? So you can do very simple analysis like measuring the nucleotide diversity as pi, and then the first answer is no, you have very low and very reduced levels of diversity in these 3 clonal lineages. And that can be suggestive of a bottleneck process. So imagine that in the center of origin, there were some samples or there were some isolates that were traveling long distances into new geographic areas, and then this is what we know as a founder effect process. So these very reduced genetic populations start like growing in these new geographic or environments. And therefore, they have a very reduced genetic variation. But most interesting is like, have these clonal lineage specific genetic diversity, despite that they have very reduced variation, have they fixed genetic diversity? And the answer is yes. So every single time that you compare, for example, here, the blue versus the green, the red versus the green, the red versus the blue, that the overall distribution of values is way higher than if you compare them, all of them, versus the orange, which is like the mixing type of individuals. By looking at this value, you can immediately see that there is some level of fixation of a specialization in these particular 3 clonal lineages. So my 2 take-home messages is yes, they have been reduced in genetic diversity, yet they have been specializing in different host environments, latitude environments, and they have been specializing as genetic lineages. And with this, I want to summarize that the feasibility of the real-time genomic monitoring with long-read sequencing technology is putting in front of us the capacity of do this type of scale type of analysis. I want to insist that population structure analysis are fundamental and are the base of understanding the evolutionary history of the lineages. And then a combination of orthogonal methods is what you want to do. You don't want to stick with a single methodology because, as you can see, they can have pros and they can have cons. So just by taking a look simultaneous and a complete picture with different orthogonal methodologies is always the way to go. Taking into account the demographic processes, the biology of the organism, the developmental stages, they shine some light and they tweak the interpretation or the results themselves. Think about the ancestry type of results. We could have suggested that there are some individuals which are a mixture of different components, but what we know is that the other way around. There is a pool of individuals that have been dispersed through different geographic regions, and therefore they are the source origin of population. So the interpretation can really shift if you put into account the biology of this particular organism. About the biology itself, what we have seen is that clonal populations are characterized by low levels of genetic diversity. Clonal populations have fixed specific allele variations. And there is the opening question: how do clonal populations thrive despite the reduced genetic diversity? This is a fascinating question that we are working on. Happy to hear your thoughts, your questions, and your comments. I want to acknowledge and I want to thank all these people that have been fundamental in many of these analyses that I have shown you, and many of the knowledge that I have been built on these type of projects, which is not just the rice, but also wheat and some other projects with this fantastic group of people. And thank you very much for listening.